This is where the story of Dr. Heimfeld's quest originates. The modern search for the hematopoietic stem cell began with the detonation of the atomic bombs over Japan in 1945. Researchers found that the intense blasts of radiation destroyed blood cells and that people often died within weeks of exposure. Scientists mimicking that exposure in mice soon realized these deadly effects could be prevented by transplanting bone marrow. They reasoned that the marrow must contain some magical cells capable of regenerating the other blood cells. Dr. Heimfeld came to Stanford University with that goal in mind, to purify those magical cells.
I got interested in science at a very young age. I've always been interested in puzzles. I like to do jigsaw puzzles and crossword puzzles. And then I got interested working with my dad on car repair and trying to figure out how things work. So my graduate work project was on Hydra. You can think of Hydra like the Hydra of Greek mythology. If you cut off its head, it'll grow a new one. My experiments were really trying to establish how that animal could repair and rejuvenate itself. And that really sparked my interest in stem cells.
When Shelly Heimfeld began his postdoctoral studies in the Irv Weizman lab, a key piece of breakthrough technology which would further his stem cell research was being developed in a lab just one floor up. Leonard Herzenberg had invented the fluorescence-activated cell sorter, or FACS, in the late 70s and had continued to develop and refine the machinery to rapidly separate cells on the basis of cell surface markers. The FACS was like a super smart kindergartner, selecting specific crayon colors from a massive box of crayons. Researchers the world over wanted access to this machine, and Shelley managed to get time with it after hours. Several times a week, Shelley would play volleyball with members of the Herzenberg lab, and eventually they trained him to run the machine himself. He often did the work for his project in the middle of the night.
One of the first things I had to work on was a pre-enrichment scheme using magnetic beads. These were small particles that we could attach to some of the cells in the bone marrow, and then using a powerful magnet remove those cells that we didn't want and keep the cells that we did. From there, we could take those cells to the FACS, to the fluorescence-activated cell sorter. This was a new technology that had been developed in the laboratory upstairs, the Herzenberg lab. And it was really a very ingenious concept of taking cells and putting them in a fluid stream, shaking the stream using an inkjet printer technology so that you could form droplets, you could charge the droplets, and sort out those cells that were in those droplets one way or another to get the ones that you wanted and get rid of the ones that you didn't want. Radiated to mice, we sorted the cells using the FACS, and then we injected them in very small numbers, 10 cells at a time, 20 cells at a time, 50, 100, trying to see what would happen when we gave those few cells. And for controls, we gave whole bone marrow that hadn't been separated, and there we had to give 100,000 cells, that we knew, in order to get the mice to survive. So then became the waiting period, and it was the Grim Reaper at that point, having to watch over the mice, come in every day and check to see who was alive and who was dead. You can think of stem cells as like a magical seed. Unlike a normal seed that only grows into one type of plant, stem cells have the property that they can make all different kinds of seeds, sort of like the different trees in a forest. Think of that as your blood.
After 2 weeks, some of the untreated mice were dying, and others were very sick. Excitement began to build as each day went by, and the stem cell-treated mice remained healthy. In fact, at the end of the experiment, all of the treated mice lived. Amazingly, mice that received as few as 100 stem cells survived. These few cells were able to keep the animals alive after a lethal dose of radiation.
So after we had all this data, now was a whole new world that was opening up. Now we had to write the paper, and there were long discussions between Jerry Spangrud, Dr. Weissman, and myself on where to submit the manuscript. And Weissman was pushing hard to get these amazing findings published in the prestigious Science Journal. This was amazing to me. That's the journal that's got only the very best and most novel findings that are accepted for publication.
Shelley's experiments opened a world of possibilities for the use of stem cells in medicine. Today, The legacy of Shelley's work can be seen in the exploration of many devastating diseases, including cancer, neurological disorders, diabetes, and heart disease.